Duchenne muscular dystrophy, also known as DMD, is a rare muscle disorder that affects 1 in every 3,500 males worldwide. DMD is caused by a mutation in the X chromosome. This mutation disrupts a gene called dystrophin, which produces the protein dystrophin. Dystrophin is a structural protein located in the sarcolemma that is involved in binding actin-based cytoskeleton to the extracellular matrix and protects the membrane from contraction-induced injury. Without this gene, this binding will not occur. The only thing present in the extracellular matrix would be a laminin. As a result, muscles will degenerate. There are multiple ideas being thrown around as to how to combat DMD, the first one being exon skipping. But to understand the purpose of exon skipping, we need to know what a reading frame is. A reading frame is how you read a sequence of nucleotides that make a series of amino acids. To start this process, you translate the DNA strands into mRNA strands, and then transcribe the mRNA into amino acids. mRNAs mostly consist of exons and amino acids grouped together to form proteins. In this example, the nucleotide sequence TAC, ATA, CGA is translated to AUG, UAU, GCU. From there, the nucleotides are grouped into triplets, which is called a codon. And for every triplet, one amino acid is produced. So in this example, the amino acids methionine, tyrosine, and alanine are produced. In the case of DMD, we will be using the reading frame shift based on the deletion of exon 50. With this deletion, exon 49 and 51 will come together. However, these two do not have similar ends, so a non-functional dystrophin protein is produced. This type of reading frame shift is known as out-of-frame error. To overcome this mutation, an antisense oligonucleotide, or an AON, can be used. An AON is used to turn off an exon, so in this instance, an AON will be used against exon 51. This results in exons 49 and 52 to combine together, and since they both have similar ends, the reading frame remains intact. This is known as an in-frame error. Although not all dystrophin protein will be produced, a truncated version will be, and that results in a milder disease known as Becker muscular dystrophy. Another idea is CRISPR-Cas9. CRISPR-Cas9 originates as a defense mechanism bacteria have against bacteriophages, or viruses that infect bacteria. Bacteriophages do this by injecting their own DNA into the bacteria. However, bacteria have developed this complex that forms when viral DNA has been detected. This complex involves a guide RNA and the nuclease Cas9. This guide RNA contains a sequence that matches the viruses, and Cas9 has the ability to cut DNA. So once the matching sequence has been found in the viral genome, Cas9 makes a cut, causing the genome to be non-functional. In terms of DMD, CRISPR-Cas9 will be used for a permanent repair to the mutation in the dystrophin gene. The last idea in this video will be the utrophin upregulation. Utrophin is an analog to dystrophin, meaning their genomic and protein structures are very similar. Utrophin is located at the neuromuscular junction in wild-type muscle. However, in the absence of dystrophin, utrophin can be found around the sarcolemma, compensating for the dystrophin deficiency. Although there is a small difference as to how the two proteins bind to actin, it has been found in clinical studies in MDX mice, or mice also missing dystrophin, to be a suitable replacement for dystrophin.